Thanks for the nice introduction, and thank you everyone for sticking around for the last session. My name is Rob Frost. I'm a faculty member in the Department of Biomedical Data Science, along with Lee here. And my first time at GLBIO, so thrilled to be here. Much hotter than I expected Minnesota to be. And excited to share some work. This is kind of a project that I've been doing on my own, independent of my trainees, and this seems like a great venue to share this at. So bear with me. Some of the math and concepts are a little complex, but hopefully I can you at least come away knowing what quaternions are, something like quaternion mathematics. So I'll start with just the motivation for why I is pursuing this, and then because I imagine most people in the audience may not be familiar with hypercomplex numbers or quaternions, I'll just review what they are, what their properties are, and why that is intriguing for these analyses, and then I'll talk about the model that I'm proposing for mapping spatial transcriptomics data to quaternions. And then once you have that quaternion model, what can you do with it? And I'll illustrate all the example applications using a Visium HD data set, which is a really exciting type, I think actually the most useful type of platform for spatial transcriptomics. So I'm sure you're all actually familiar with this really exciting area of spatial transcriptomics and single cell transcriptomics. And over the last 10 years, we've seen just dramatic changes in the underlying technologies. And although the model I'm going to present is relevant for both single-cell RNA-seq and spatial transcriptomics, I'm particularly interested in spatial transcriptomics, treating it essentially as like an image, so we can do two-dimensional based Fourier analysis of it. And this slide, you can't probably really see it, but it's a review that's kind of highlighting the two main types of technologies here. And so on the top, we have the sequencing-based ones, and Visium, the 10x Visium system is the key example here, and these have the benefit that they are unbiased, so you get effectively genome-wide measurements. But the disadvantage is that they're on a grid, and the grid may not align to individual cells, and so you could often have this deconvolution or cell mapping problem. And the key alternate technology are the imaging-based ones, and these are probe-based. And so 10x Xenium is the key example here. And you get now, actually with a 5K, like roughly 5,000 plus some custom genes, but it still has some limitations. The key advantage is its subcellular resolution. So you can actually segment cells and map counts to cells more accurately. The problem with both of them is they generate enormous matrices. And so anyone who has done like the old Visium, which was 5,000 spots, now you're getting 1.2 million 2-micron spots. So there are all of these complex deep learning methods that people developed on Visium data that just do not run at all on Visium HD or on Xenium data. And in order to be tractable, you usually have a series of approximations. So people have to reduce the number of genes through feature selection, then they reduce the number of locations from sketching, and then on this sketched feature selected data, they then perform dimensionality reduction. And they have to do all of that before you can do any kind of downstream analysis like clustering. In my own research, I'm most interested in the dimensionality reduction part of this. And of course, like the standard is usually truncated PCA followed by maybe a nonlinear method like UMAP for visualization. You can also do this in a supervised context by kind of treating like gene set testing. We'll kind of aggregate genes by biological function. And what I want to do, well, I really wanted to explore hypercomplex numbers and quaternions, but I want to explore a very different type of dimensionality reduction, where you take some 4-dimensional latent space and you map it onto individual numbers. So each location is going to be represented by one number, a quaternion, which is this 4-dimensional hypercomplex number. And what I really wanted to be able to do is to turn the data into a matrix where the rows and columns are the X and Y coordinates in the tissue, and it's a matrix of just one number, these quaternions. And then you can treat your spatial transcriptomics data like an image, do all these image analysis methods on it. Okay, so what are quaternions? So they are one of the 4 hypercomplex algebras that are the 4 normed division algebras. So they're of interest to pure mathematicians. But of course, in an applied space, we deal often with the first 2, like real numbers and complex numbers. And the way to think of quaternions is they are a generalization of complex numbers where the imaginary space goes from one component to a 3-dimensional imaginary space with I, J, and K. These are all square roots of -1. And some operations on quaternions are straightforward. So adding them is pretty straightforward. Scalar multiplication is pretty straightforward. You just have the expected outcomes. It's multiplication and division that become complex because this is a non-commutative algebra. So I times j is k, j times I is -k. And for people who have more of like remember your physics and vector analysis, this acts a lot like a cross product, the right-hand rule, positive or negative. And this is the one thing where they're actually useful in practice currently is modeling rotations because of this non-commutative nature. So we can define a conjugate in the same way we do for complex number, and we can define a norm. And with the conjugate and norm, we can define a reciprocal, and then we can define division, so the quotient. And because it's non-commutative, you actually have left and right-hand versions of this. It's often more useful to represent this in this notation of a real component and the vector component. And I'll talk a lot about this vector component, which is the 3 imaginary dimensions. And using that notation, we can restate quaternion multiplication using that form, which is arguably more interpretable, but still a challenge. So it becomes this function of the product of the reals, the dot product of the vectors, the scalar multiplications, and then a cross product. And so it is not, this is the one challenge with quaternions, it's not intuitive often how to interpret things. But it helps to think of 2 special cases. So if one of the quaternions is just a real number, this simplifies the scalar multiplication, so it's pretty straightforward. If they are both vectors, so they have no real component, then we get this real component becomes the dot product of the 2, and the imaginary component becomes a cross product. So if you have 2 orthogonal vector components, you end up with just another vector component. You stay purely in the imaginary space, and either the right-hand rule kind of applies. And if they're both, if they're parallel, then the imaginary component disappears, and you end up with a real number, which is the dot product. So how to use, what's the utility for these? And the main area where they have been practically used, and you probably use them all the time in just a 3-dimensional graphics library on your computer, is modeling rotations. So you can just represent a vector in 3D as a vector quaternion. And then they have this really nice property that you can define any rotation about an axis and an angle with another quaternion called the rotation quaternion. And then you compute the output of that by pre and post multiplying by that quaternion. And you will end up there without a real component and just another vector quaternion. And it has some benefits relative. You would normally think of doing 3D rotations using a rotation matrix. You need 1 quaternion, so you only need 4 numbers to describe the rotation versus 9 for a rotation matrix. And the argument is that it's more straightforward. So it's more easy to go from the vector quaternion to your axis of rotation and the rotation angle. But the key benefit is if you have a whole sequence of smooth rotations you want to compute, it's much more efficient and numerically stable to do it with vector quaternion. So this is how they mainly get used outside of pure math. Okay, so that's what quaternions are. This is we can represent 4 numbers, it's 4-dimensional space with each one. They're really good for rotations. How to map spatial transcriptomics data to quaternions. And then once you have them, what do you do? What can you do with it? So there are 2, in the paper, I propose 2 different models. So one I'm going to call the singular value decomposition model, and the other is the gene model. So for the SVD model, you just start with your unnormalized counts, and you just compute a truncated SVD on that. And I generally use like a randomized truncated SVD for efficiency. And you can think of this as an uncentered principal component analysis. And then for every location in your data set, you're going to create a quaternion. And the real part will be the absolute value of the projection onto the first uncentered pre-C. You compute the Euclidean length of the projection onto the other 3. And then you create basically a unit length imaginary vector. So the vector quaternion is this unit length vector spanning the uncentered principal components 2 through 4. And then with all those quaternions, you can put them into a matrix. There's actually an R package that supports matrix efficient things, and I'll mention that later on quaternions, and actually the 8-dimensional space as well. And the rows and columns of this matrix, we think of our data as the rows being the cells or locations, and the columns being the genes. But here now our matrix is actually going to represent the 2-dimensional geometry of the tissue. So they're going to be the unique X and Y coordinates, and every element will be a quaternion. And this has a nice property that the real component captures library size. So uncertainty in your relative expression profile. And the vector component captures the relative expression profile, or an approximation of it. And this is just some simulated data to just illustrate this fact. One nice feature of this is that you can think of rotations of the vector quaternion, because this vector quaternion part is the relative expression profile. Rotations of it are like changes in transcriptomic state, so the phenotype or state of a cell. And the rotation itself is described by a relative expression profile, the axis. That's another quaternion. And you could think of that, then the rotation axis is some relative expression profile that ideally, if this holds, is invariant to the change. So it has this kind of biological interpretation of rotations. So you can do this. You don't have to do this for kind of an SVD-based kind of latent space model. You can do it for target genes. The examples I'm going to show are actually reduced rank reconstructed genes. You can do it for gene sets or some other variable, and it's an analogous mapping. You're going to compute a kind of Euclidean length over them. You set the real component to the length, so it's the approximating library size, and then you create a unit length imaginary vector. So there are a number of things I think that are interesting to do once you've turned your spatial transcriptomics data now into this matrix of quaternions. Like, what can you do with that? And there are 5 I'm going to show you. And the first 2, so one's like a visualization of this latent space. The second is doing rotation-based analyses. And I put asterisks by those because you don't technically need quaternions for these. But I think, especially for the rotation analysis, it's more efficient. It has a nice interpretation when we're doing it. The last 3 are all Fourier-based. So this is 2-dimensional, hypercomplex domain Fourier transformation-based analyses. And for these, you do need quaternions. And so all the analyses I'm going to show, there's an R package that's associated with the paper. And it depends on, there's an onion R package, which actually supports hypercomplex computations in R. And the randomized SVD that I use is RSVD package, and then Seurat for all of the spatial transcriptomics analysis. Okay, so we're now going to create these matrices of quaternions, and I'll talk to you what you can do, those 5 applications, using a Visium HD data set. And this is actually for people who do spatial transcriptomics, this is the one I'm most excited about. So we went from Visium, which was old and had these like 55 micron spots that were spaced out. You get like 1 to 10 cells, and they were not actually that useful. It was kind of novel. And a lot of my collaborators have been really excited for Xenium, but then they're generating this Xenium data on only like 300 genes or 500 genes. It turns out there's not enough. Whereas Visium HD is, in theory, genome-wide. And I actually think this is the most potentially useful form. So this is just a visualization of this. It's a mouse brain kernel section on an FFPE tissue. And this is colored according to library size. So you actually get 1.2 million 2-micron grids, but you usually aggregate that for sparsity to 8 micron or 16. And so I aggregated this up to 16, and it still generates 100,000 non-empty locations. So it's a big data set. And then I'm creating from this 2 different matrices of quaternions, and they're both 368 by 465, which is the number of unique XY coordinates on the tissue. And I'm not directly, so for the gene-based model, I'm using a GFAP renin neuron 6. So these are markers for astrocytes, interneurons, and excitatory neurons. And rather than directly using the gene values, I'm using the reduced rank reconstructed values, which works much better for this data. And okay, this is the first application. This is actually visualizations of those quaternion matrices. And because they're 4-dimensional, you have this nice mapping from quaternion coefficients to RGB plus opacity alpha values. And I think these make kind of strikingly beautiful images of the data. And this is not so novel, right? This is just a false color representation of a 4-dimensional latent space. I don't see this done actually that often for spatial transcriptomics data, representing this reduced dimension version. But on the gene-based model, it's a clear interpretation. The red are predominantly areas with astrocytes, the green interneurons, and the blue excitatory neurons. Okay, so one of the first analyses was the rotation. And so this is actually which is very easy to compute with quaternions. This is a rotation about the IJ quaternion axis, a pi radian rotation. And so it makes a nice, beautiful image, but it's a little challenging to interpret. So I think it's easier actually to look at the Euclidean length of the vector component of the difference between the unrotated and the rotated matrices. And the interpretation here is that if the spot, essentially the quaternion for the spot is orthogonal to the axis of rotation, it won't change the length, the difference. And if it's very close, the difference will have a very short length. And so it just highlights what we'd expect the IJ or the astrocytes and interneurons. So on the right-hand side, the darker things with the larger lengths are the areas that are predominantly excitatory neurons. So this, I think, is a more intuitive and nice kind of useful visualization of this. Okay, onto the Fourier transform-based ones. So of course, one thing we can do is we can do like Fourier-based filtering. And so this just illustrates the output of a low pass or high pass filter. And the important thing is with quaternions, of course, we can do two-dimensional, FFT-based discrete Fourier transforms on real numbers, matrices of real numbers or matrices of complex numbers. But you can also do it. There's a way of mapping the quaternion one to complex domain, two different complex domain Fourier transforms, which is the kind of key benefit. I don't know that this is very biologically helpful, but it's just like proof of principle. We can do it. This, I think, is actually more useful. So another thing that Fourier transforms are great for is convolutions. And this is showing the convolution with a masked matrix that embeds a edge detection kernel. In this case, it's like a real value kernel. And I think it's for the SVD-based model, it really kind of beautifully highlights boundaries between different biologically relevant tissue domains. So it's easy to do the convolution with a real value kernel. It is very challenging to design and interpret convolution with a single mask matrix that's quaternion valued because it's non-commutative. So that is a challenge. And one thing that other people in the past have explored for doing this with other kind of quaternion models is a biconvolution, where you think about this quaternion rotation, you're pre and post multiplying so that you kind of eliminate the real component. And there are ways of doing effectively edge detection in a space orthogonal to an axis of rotation. And that's what this is showing for those two. Okay, so just in conclusion, so this paper outlined a novel, and I would say it's still very much kind of proof of concept exploratory mapping of transcriptomics data, specifically spatial transcriptomics to quaternions. And there are two main benefits. One, it allows you to turn your spatial transcriptomics data, this kind of a approximate reduced dimensional representation of it, into a matrix of numbers, quaternions, and then you can do all these Fourier-based analyses on that. And I think it has also this nice interpretation of rotations. But it does have some key limitations. So they're four-dimensional hypercomplex numbers. That's all you get is four values. And so this is really just a rank 3 embedding of the normalized data. You could say you're missing a lot of biology. But of course, look at all the figures in papers that are doing single-cell RNA-seq. They're having a UMAP plot. They're showing all this biology just in 2 dimensions. So I think if what you care about is capturing the main cell type or subtype groupings, then that's realistic. And they're also challenging to interpret, right? They're non-commutative. And so interpreting and validating it is a challenge. So my future work is mainly trying to explore the practical applications in benchmarking and comparative analyses to try to overcome some of those limitations. And just acknowledge my grants that funded this. And thank you, and happy to take any questions.
Hi. So I'm trying to think through how applicable this would be to mass spec imaging, because you talked about the real component being representative of the library size, but the sum of the pixel intensity in mass spec imaging isn't super meaningful. So would I have to do some modification to this procedure to just port that over?
That's a great question. I haven't thought about it and would love to talk to you more about the mass spec. I mean, one way of doing it, you can just ignore the real component and map a 3-dimensional space to the vector part of the quaternions. If you don't have some measure, I think using library size for the real part made it nice to have interpretation of the opacity parameter. So we have, for areas that are more uncertain, they become more transparent. In areas that are less uncertain, and then you can also visualize the uncertainty interval as an expression profile. But so one way is just to ignore the real part and just focus on the vector part of it, the 3-dimensional mapping.
If the real part its only sort of useful interpretation is just that opacity, could you just add a fourth dimension in there and say, hey, I've got higher dimensional data now?
You can, but then some of the interpretation of rotations changes in terms of, yeah, so it becomes challenging, I think, to just simply use the same essentially types of latent variables for both the imaginary and the real one. That has some other interpretation challenges, but great question.
Okay, cool.
Thanks, Aaron.
Thank you.
Yeah.
Interesting.
Have you tried applying your method to Cosmix data from the NanoString platform? Cosmix data? Have you tried applying it to
No,
that?
I have not.
Okay.
Um,
It would be very interesting to see how that works with that data set. That's one of the platforms that a lot of us are using now.
Oh, cool. Thank you. Thanks for the suggestion. Yeah, is any, and happy if people are curious about this later. If anyone wants to try it out, I'm happy to talk you through using the R package.
Right, another round of